Today I'm answering this GCSE AQA Biology paper. It's a higher tier paper from June 21. A student prepared some animal cells to view using a microscope. Figure 1 shows the student preparing the cells. Name 2 pieces of laboratory equipment the student could have used to prepare the cells to view using a microscope. So we have some animal cells. What do we need to do with them? Well, we need to pop them onto a slide and then place a cover slip over the top. And that's to keep it in place. You could have also mentioned a mounted needle. You could have used a pipette, a scalpel, forceps, tweezers, dye, stain, lots of options. Figure 2 shows the student's light microscope. Name part A. This is where you put your eye. Remember, it's up here. So that is the eyepiece. You could have also written lens. What is the function of B? So remember, when you turn this, it actually enables you to focus on the cells on your slide. The student tried to look at the cells using the microscope. Suggests one reason why the student could not see any cells when looking through part A. Maybe the light wasn't switched on after all. It is a light microscope. However, there are lots of options. You could have said that potentially there were no cells in the field of view, that the slide wasn't in the correct position, the lens was dirty, the microscope wasn't focused. Red blood cells are specialised animal cells. Compare the structure of a red blood cell with the structure of a plant cell. Okay, I'm just going to draw a red blood cell up here. Remember, it's got that characteristic biconcave disc shape. Weirdly, it has no nucleus, which is unusual for an animal cell, but that's to enable it to have more hemoglobin. If I draw a plant cell up here, it's got a different shape. It does have a nucleus. The main difference between this and the red blood cell is that the plant cell has a cell wall. However, there are lots of similarities, such as the fact that they both have cytoplasm, cell membrane. So we're now going to compare these two cells, saying what's the same and what's different. Because after all, that's what compare means. Now we're going to consider the differences. So let's point out the main differences. First of all, between a plant and animal cell is that the plant cell has a cell wall, chloroplast and vacuole. Next up, we're talking about the shape, whereas the plant cell is rectangular. And lastly, the red blood cell has hemoglobin. I'll just highlight where the marking points are. So you get a mark for each of these, the fact that they both have a cell membrane and a cytoplasm. This comparison of chloroplast is good. The vacuole, the nucleus and the cell wall, those are all worth a mark. The shape is worth a mark. And the fact that the red blood cell has hemoglobin. So I've well and truly hit the 6 mark point. When placed into a beaker of water, a red blood cell bursts. A plant cell does not burst. Explain why the red blood cell bursts but the plant cell doesn't. This is about osmosis. It's worth 2 marks. So why does the red blood cell burst? Because if you think about the difference in structure, the red blood cell has no cell wall. The plant cell with a cell wall cannot burst, even when the water enters. A student investigated the effectiveness of 3 different antibiotics. Remember, antibiotics kill bacteria. Figure 3 shows how the student set up an agar plate. The student used aseptic techniques to make sure that only one type of bacteria is growing on the agar. Describe 2 aseptic techniques the student should have used. So, how are we making sure that we have sterile conditions? Well, when we add bacteria to the agar jelly, we need to use a sterile inoculating loop. The other thing you want to do is make sure that when you've added your antibiotics, you place the lid on and seal it with tape. It's raining if you can hear that. You could also mention only lifting up the petri dish lid a little bit, or you could have used sterilised agar. The student placed the agar plate in an incubator for 48 hours. Figure 4 shows the agar plate after 48 hours. Look, here's your area where no bacteria are growing. So actually, this is the best one, the best antibiotic. This one's the worst. Why? Because you've got the biggest clearance zone here, which means all the bacteria have been killed here. Very few have been killed there. Which antibiotic is the least effective? Give a reason for your answer. I've already labelled it. It's B. Why? The clearance zone around the antibiotic is the smallest. Calculate the area where no bacteria growing for antibiotic C. Use pi equals 3.14 and give the unit. So we want to measure the area of this circle using pi r squared. So measure this distance here using your ruler. Let's measure it in millimetres. So our radius is 11 millimetres. And so therefore we do area equals pi, which was 3.14, they told us in the question, times 11 squared to get 379.94 millimetres squared. Area equals pi r squared. Pi was 3.14, r was 11 millimetres. The answer here is 380 millimetres squared to two significant figures. You could have also done it in terms of centimetres, but I prefer to do it in millimetres personally. Suggest one way the student could improve the investigation. The simplest way always is to repeat and calculate an average, because remember, that improves the reliability. Body mass index, BMI, is a way of finding out if a person's body mass falls within a healthy range for their height. Table 1 shows information about 2 people. Make sure you read every single word. Figure 5 shows 5 BMI categories for adults. Which is the BMI category for person A in Table 1? Let's make sure we're looking at the right row. So we know that their body mass is 63. So we need to go up here. And we know that the height is 1.65. And just look for where that crosses over. It's in this light grey section, so we're going to say normal. Calculate value x in Table 1 and use this equation. Give your answer to 3 sig fig. So now we're looking along this row. We have a body mass of 92 kilos, height is 1.71. We need to square that, 31.46. But we want it to 3 significant figures, which means we only want 3 numbers. So look at your 4th number, it's above 5, so we round up. That's 31.5. Scientists think there is a link between BMI and life expectancy. Table 2 shows information about the predicted life expectancy of men after the age of 50. Describe 2 patterns shown in Table 2 about the effects of the BMI category. You can see here that the higher the BMI category, the lower the number of years of living in good health. The higher the BMI category, the higher the numbers of years living in bad health. So you're just saying what you see from the data, no scientific explanation if you're asked to describe. The number of people who are obese in the UK is increasing. Explain the financial impact on the UK economy of an increasing number of people who are obese. So the UK economy will be adversely affected, be weakened, be less strong. Why? Because there are fewer healthy people able to work. person who is obese is at more risk of arthritis. Arthritis is a condition that damages joints. Suggests how arthritis could affect a person's lifestyle. They'll be less able to move. A person who eats a diet high in saturated fat might become obese. Name 2 health conditions that might develop if a person eats a diet high in saturated fat. Do not mention arthritis. Why? Because they've just told us that. So you might get type 2 diabetes or you might get coronary heart disease. It is important that you mention type 2 because type 1, remember, has a genetic cause. All living organisms respire. What is the chemical equation for aerobic respiration? Aerobic meaning there's a requirement of oxygen. You do need to learn this equation. It produces carbon dioxide and water. So let's look down here and see what we can find. Okay, well there's oxygen and carbon dioxide. So we definitely know it's not that. This is water and glucose. Again, not what I've written. Next up we have water and carbon dioxide. No. But lastly, we have oxygen and glucose forming water and carbon dioxide. It is this one. Name the subcellular structures where aerobic respiration takes place. Remember that takes place in the mitochondria. Energy is released in respiration. Give 2 uses of the energy released in respiration. Well, that energy allows our muscles to contract. It also enables us to build large molecules from small molecules. You could also say it helps us keep warm. It enables us to carry out active transport of substances within our bodies, as well as cell division. Describe 2 differences between aerobic and anaerobic respiration. Humans do not refer to oxygen in your answer, so read every word. Aerobic respiration, remember, is a lot more efficient and produces more ATP compared with anaerobic respiration, or you can say it releases more energy. What about byproducts? Well, anaerobic respiration is famous for producing lactic acid, which is why our muscles feel so fatigued and tired. What are the 2 products of anaerobic respiration in plant cells? Don't be tempted to pick lactic acid because remember that's to do with animal cells. Remember that anaerobic respiration in plant cells produces both ethanol and carbon dioxide. A scientist investigated respiration and photosynthesis using some pondweed and a pond snail. Figure 6 shows the apparatus used. The apparatus was left in a well-lit room for 5 days. That's important because it means that the pondweed can photosynthesize. The data logger recorded the concentration of carbon dioxide continuously. After 5 days, the scientists completely covered the boiling tube with black paper. The data logger continued to record the concentration of carbon dioxide. Figure 7 shows the concentration of carbon dioxide inside the boiling tube over the 15 days. So just so you know what's going on, because here it was covered in black paper, it means that there's no photosynthesis, which means no absorption of carbon dioxide in that process, which is why we can see increased carbon dioxide over here because of all the respiration taking place, because remember that doesn't require light. And then on this side, lots of photosynthesis taking place, meaning decreased carbon dioxide, and you can actually see that it's much lower compared with this side. Explain why the concentration of carbon dioxide in the tube stayed the same between day 0 and day 5. So from here to here, it's because the pondweed is actually taking in that carbon dioxide for the photosynthesis, but then the pondweed and the snail are both respiring, which releases carbon dioxide. So the neatest way of saying that really is that the rate of respiration equals the rate of photosynthesis. Pondweed is photosynthesizing. Pondweed and snail are both respiring. Suggest why the concentration of carbon dioxide increased between day 5 and day 10. So here, mention this because the pondweed is no longer photosynthesizing. but both the pondweed and snail are still respiring. On day 10, the pond snail died. That's horrible. Explain why the death of the pond snail caused the concentration of carbon dioxide to increase after day 10. So what would benefit from the death of the pond snail? Bacteria feeding on it, which are also respiring and therefore releasing carbon dioxide. So the snail is decomposed by bacteria. Bacteria respire, releasing carbon dioxide. And that is enough for 3 marks. Amylase is an enzyme that breaks down starch. Amylase is a polymer of smaller molecules. Name the type of smaller molecule. Be careful here. Remember that amylase is an enzyme, and an enzyme is made out of protein. So if you think of it as a protein, what are the small subunits known as? They are amino acids. So this is really checking that you understand that enzymes are made out of protein and therefore made out of amino acids. Name 3 parts of the human digestive system that produce amylase. That is the salivary glands, pancreas, and the small intestine. Explain how amylase breaks down starch. Answer in terms of the lock and key theory. So if we draw a picture, here's your amylase enzyme. It has a special part on it called the active site, which, remember, binds to the substrate, the starch. The reason we think it's a lock and key is because, after all, the enzyme's active site is complementary to the shape of that substrate starch. So let's write that. Starch binds to amylase's active site. The shape of the substrate, which, remember, is the starch, is complementary to the enzyme's active site. And then lastly, what happens, because it said how does amylase break down starch? Starch is digested into maltose. A student investigated the effect of temperature on the activity of amylase. So that is our independent variable, what we're changing. Figure 8 shows the apparatus used. This is the method used. Set up the apparatus as shown in Figure 8. After 5 minutes, pour the starch solution into the amylase solution and mix. Remove 1 drop of the starch amylase mixture and place on a spotting tile. Add 2 drops of iodine to the mixture on the spotting tile. Record the colour. Repeat steps 3 to 5 every minute until the iodine solution stays yellow-brown. Repeat steps 1 to 6 using different temperatures. A few things to point out. Remember that starch turns iodine blue-black. And that means if starch has been broken down into maltose, maltose will not turn iodine blue-black. Name 2 control variables that the student used in the investigation. So what did they keep the same? The volume of starch solution and the volume of amylase solution. You could have also said the volume of iodine added. Why did the student leave the starch solution and amylase solution for 5 minutes before mixing them? That's simply to allow the solutions to reach the same temperature, which is 5 degrees. Table 3 shows the results of the investigation. Here we're saying time taken until the iodine solution stays yellow-brown in minutes. This one's actually happened the fastest here. So for some reason 35 degrees is the best. What conclusion can be made about the effective temperature on amylase activity between 20 degrees and 65 degrees? Well, we can see that as the temperature increases, the time taken decreases, which means that amylase's activity is much higher, much greater. Above 35 degrees, we can see the time taken increases, which means that the amylase is not working as well. And then above 35 degrees, amylase activity decreases. Explain the results at 5 degrees and 80 degrees. So we need a scientific reason here. And it's worth a lot of marks, 5 marks. Look, the results are the same, but there's a different explanation for these. So why might 5 degrees mean that the iodine didn't turn yellow-brown? Because the amylase is too cold, meaning it has low kinetic energy, and therefore it's not likely to be catalyzing the reaction. It's not breaking down the starch. But up here we have a different reason. Although again it's working extremely slowly, why is that? Because remember, high temperatures, the amylase enzyme is denatured, and we're going to say exactly what that means. So at 5 degrees, amylase has low kinetic energy, therefore few collisions between the enzyme amylase and the substrate starch. Then we'll make a statement at 80 degrees, amylase is denatured. What does that mean? Its active site has changed shape and the starch no longer fits, or the substrate. Remember, these words are interchangeable. Poor amylase activity is seen by the iodine remaining orange-brown, meaning that the starch hasn't been broken down. The student investigated the effect of temperature on amylase activity. Describe how the student could extend the investigation to determine the effect of a different factor on amylase activity. Well, we could look at pH and what effect that has. So use a range of different pHs. But remember, we need only one independent variable, so we'd have to keep the temperature the same. Figure 9 shows a cross-section of a leaf. Which cell is most transparent? So this is the waxy cuticle up here. Then you have the upper epidermis, which is transparent to allow light to reach the palisade layer down here. So that's why it's A. Which cell structure in a leaf mesophyll is not found in a root hair cell? So what's different about a leaf and a root? Well, a leaf wants to photosynthesize. A root can't do that because it has no access to light. So what would be the major difference in the organelles found? Well, the leaf cell contains chloroplast. Unlike the root hair cell. Plants lose water through their leaves. Name the cells in a leaf that control the rate of water loss. It is the guard cells. Because remember, these are the guard cells down here, and they control water leaving the stoma. So they're really letter D. Water is taken in by the roots and transported up to the plant and lost from leaves. Which scientific term describes this movement of water? This exact movement is known as transpiration stream because the attraction of those water molecules within the xylem and their loss from the leaf helps draw more water up from below, which is why it's called a stream, which change would decrease the rate in water loss from a plant's leaf. To decrease, we want it to be humid, we want it to be cold, we want it to be dull outside, so not sunny, and we want it to be not windy. So what have we got here? Yep, that's one I've said, so that's why that's the answer. Compare the structure and function of xylem tissue and phloem tissue. Xylem tissue is dead, phloem is living. Let's talk more about the structure. Xylem has lignin to help support and hollow tubes to enable the transport of water and mineral ions, so we've touched on the function there. Let's make a comparative statement. Phloem does not have lignin. Instead, it has sieve tube elements in cytoplasm that enable the transport of sugar. Let's talk about direction of movement. Remember, xylem transports water just upwards from the roots. Phloem transport occurs in both directions. Both xylem and phloem consist of cells. That is such a bland point. Figure 10 shows the total volume of water lost from a plant over 6 hours. Determine the rate of loss of water at 12 o'clock. Use the tangent on figure 10. Give your answer in centimetres cubed per minute and in standard form. Okay, so here's our tangent. It's nice that they've drawn it for us. Because it's a rate, we want to do change in y over change in x. Let's keep our units in check as well. So change in y, let's look from here, 190 to 230 is 40. Our unit there is centimetres cubed. Change in x. So we're going from 8 o'clock to 3 o'clock. Make sure you count that really carefully. 9, 10, 11, 12, 1, 2, 3. That is 7 hours. Keep your units in check because remember they want the answer in terms of minutes. So we need to convert 7 hours into minutes by multiplying by 60 to get 420 minutes. So I'm happy that we're in the right unit now. Put that into your calculator to get 0.095238, and then they wanted the answer in standard form. So we do 9.5 times 10 to the count 1, 2 to the minus 2. And I've used my tangent, I've looked at the right time, my units are correct, and I'm in standard form, so I've got those 4 marks. The rate of water loss at midnight was much lower than at 12 o'clock. Explain why. So water loss occurs through the stomata. Now the stomata open to allow CO2 in, so photosynthesis takes place inside the leaf. So why stomata close? As no photosynthesis is taking place. Figure 11 shows where 3 of the same tumour were found in a patient. Malignant tumours are cancers. Describe what happens to cells when a tumour forms. It's basically saying how does a tumour form? Well, cells divide uncontrollably. What evidence is then figure 11 to suggest that the tumour in the lung is malignant? A malignant tumour is one which spreads. So it's the fact that it's spread to other parts of the body, including the liver down here. Some types of cancer can cause the number of blood components in a person's body to fall to a dangerously low level. A person with one of these types of cancer may experience symptoms such as tiredness, frequent infections, bleeding that will not stop after the skin is cut. Explain how a very low number of blood components in the body cause these symptoms. Okay, let's talk about them. Tiredness will be because you have fewer red blood cells, meaning that you have less hemoglobin transporting oxygen. You're effectively carrying out less respiration. Frequent infections will be due to decreased phagocyte and lymphocytes. So remember, these are white blood cells to do with fighting pathogens. Bleeding that will not stop after the skin is cut. Well, it's platelets, which remember are found in the blood. They're what clot the blood at the site of a wound. So fewer platelets would mean you'd get more bleeding. Let's go through these in turn. Tiredness is due to decreased red blood cells, meaning less hemoglobin and less oxygen transported. Respiration can no longer take place as much. Let's talk about the frequent infections due to decreased phagocytes, so less engulfing of pathogens, because remember that's what phagocytes do. You'll also have fewer lymphocytes, and remember lymphocytes release antibodies, and antibodies again fight pathogens. And then lastly, this thing about the skin being cut and continuing to bleed, fewer platelets to clot the blood at the site of a wound. So just making sure you're happy with this question. We know that we're talking about blood components. What are these blood components that we've picked out? Red blood cells, and then talked about their role. Phagocytes and lymphocytes, which remember are white blood cells found within the blood, and why reduction in their numbers increases infections. And then lastly, platelets, which are small fragments of cells found in the blood, which are responsible for blood clotting. Let's highlight the marking points. Fewer red blood cells is a mark, less hemoglobin is a mark, less oxygen transported is a mark, respiration no longer taking place is a mark, 4 marks. Decreased phagocytes is a mark, fewer antibodies being made is a mark, fewer pathogens being killed is a mark, fewer platelets is a mark, and this thing about clotting the blood is a mark. So we've got loads and loads of points. Some patients with very low number of blood cells may be given a blood transfusion. A blood transfusion is where a patient receives blood from a donor. Different people have different blood groups. Figure 12 shows the red blood cells found in people with different blood groups, the antibodies that can be made by people with different blood groups. So here's the red blood cell. They have little protein markers on them known as antigens, and the antibodies here will attack B antigens. Antibodies can bind to antigens that have complementary shapes. When antibodies bind to the antigens on red blood cells, many red blood cells begin to clump together. Each red blood cell has an 8 micrometer diameter. Many capillaries have an internal diameter of 10 micrometers. In one type of blood transfusion, only red blood cells from a donor are transferred to the patient. It is dangerous for a patient with blood group A to receive cells from a donor with blood group B. Explain why. So in this question, we've been told that the person has blood group A, but they're receiving blood group B blood. Why is that going to be a problem? Well, that's because the recipient person's blood will have anti-B antibodies in it. And so as that blood comes in From the blood group B donor, those anti-B antibodies will bind to the type B antigens, causing the red blood cells to clump together. And they gave us some information about the diameters. So what that will do is it will block the capillaries, meaning that the oxygenated red blood cells can no longer reach those respiring cells. So you're going to have a lot of knock-on effects there. Explain why blood group O red blood cells can be given to patients with any blood group. Awesome. There are no antigens on the red blood cells of blood group O for either blood group A or B people to respond to. Table 4 shows some of the risks associated with blood transfusions. Which risk has the lowest probability of occurring? So you're really looking for the lowest number here. Hopefully you can see by far that this is the smallest number, so it's hepatitis C infection. A person has a tumour blocking the tube leading from the gallbladder to the small intestine. Explain why this person would have difficulty digesting fat. So what is going on here? Well, the gallbladder, remember, stores bile. And why is bile important for digesting fat? Well, bile emulsifies fats. So this is going to underpin our whole answer. Bile can no longer reach the small intestine. Meaning it can no longer emulsify fats. Let's point out what emulsification is. Remember, it's taking large fat droplets and breaking them down into small ones. Smaller fat droplets no longer made, so surface area to volume ratio remains small. So the enzyme lipase cannot work as effectively. What was the other role of bile? Well, bile provides the optimum pH for the enzyme lipase. So if we don't have bile, then we no longer have the optimum pH. So pH of small intestine no longer neutralized, meaning lipase cannot break down fat as easily.